Hi, everyone. Welcome to Data Wednesdays. So this week, we are actually hosting a Brown faculty member, Professor Brenda Rubenstein, who will be giving her CCNB affiliate talk. So with that, I will hand it off to Soren to introduce her.
Hello, everybody. Hello, Brenda. I'm excited to introduce Professor Brenda Rubenstein from our chemistry department, giving a talk today on the beautiful topic of protein folding. So Brenda is the Joukowsky Family Assistant Professor of Chemistry at Brown, and she received her bachelor degree in chemical physics and applied mathematics from Brown University. Her master in philosophy in computational chemistry while a Churchill Scholar at University of Cambridge, and her PhD in chemical physics from Columbia. Her work at Brown was recognized by exciting distinctions. She was named the Alfred Sloan Fellow, a Kotler Teacher Scholar, a Camille Dreyfus Teacher Scholar, and an Air Force Young Investigator. So we are delighted to have Brenda with us today. And I'm going to give it to Brenda. Thank you.
Thank you, Soren. Thank you for the introduction, and thank you, everyone, for hosting me here today. I want to give you a particular thank you because it's so gorgeous outside. I know I was on the Cape trying to see my parents for the first time in about 2 years, and you pulled me back. So I give you a lot of credit for also coming back for this talk today. So today I'm going to talk about some of the work that's been going on in my group, which I largely classify as predicting the viability of different organisms based upon their folding, their protein folding, their binding, and their dynamics. So traditionally in computational biology, people are interested in trying to predict the fitness of an organism. So maybe how well that organism will reproduce, maybe what functions that organism will have, maybe the pathogenicity of different proteins in that organism, based upon the sequences of the proteins within that organism. However, what my group has been trying to do and trying to understand is what if we go beyond this sequence-based paradigm, right? So instead of actually mapping from DNA to fitness, why don't we start thinking about biophysical metrics? So things like folding free energies, binding free energies, protein dynamics, and seeing how well those things also correlate with fitness. And so what we're trying to do is basically dig under the hood of what all those genetic sequences mean to see if we can find better correlates with fitness than just those genetic measures. And I'll cut to the chase just for now. And the answer is that what we essentially find is that in many cases, genetic sequences do an amazing job at predicting fitness. But if we want to understand the specific details of why some organisms' fitness decreases, then it becomes important to actually look at these measures under the hood. And so I'll give you some examples today, and I'll show you, when biophysical measures work, when they don't work, and when they're sort of blah in the middle. So before I begin, for those of you who are unfamiliar with me and my work, I would say my group focuses on three key areas. All of these areas are united by their use of computational science to try to solve demanding problems in the physical and biological sciences. The first focus area of our work is on quantum statistical mechanics, and this is probably what, if you ask random people in the community what I'm known for, they would say it's this. And what do I mean by that? What we try to do in the group is we try to essentially solve the Schrodinger equation faster and more accurately than other methods. And the way we do that is actually by using statistics. So we use random samples of things like a system's wave function or other properties of a quantum system in order to try to determine what's going on in that system. Beyond that focus, we're also interested in things like alternative computation. So we've been doing a great deal of work on molecular computation. And the idea there is that we actually want to get molecules to compute for you. So historically, people have thought about molecular computation as meaning that you dial up on your computer what a molecule is doing. We actually mean the opposite. We've been working on ways to take molecules and make them perform everyday computational functions for us. We've also been doing a lot of quantum computation and biological computation using biofilms, and I'll talk about that towards the end as well. But the last area, and the area that I'm going to focus on right now, is really computational biophysics. And here, what we're very much interested in, just as I said before, is trying to predict the fitness and the evolutionary fitness landscape of different proteins, just based upon measures of biophysics. So many of you might say, that kind of sounds strange. I'm in a chemistry department. Why would I be thinking about these different things? Well, the honest answer is that this is both a new and an old passion. So years ago, I was a Brown undergraduate, as Soren said. And when I was a Brown undergraduate, I was actually a biochemistry concentrator for most of my time there. But basically, by the end of my sophomore year, after taking lots of biochemistry classes that I never got put on my degree, but I'm sure I could have gotten a biochemistry concentration, I decided I was much more interested in computation. As the story goes, way back then, we really didn't have something like CCMB. We really didn't have lots of computational science at Brown. There were things like fluid dynamics. And so I never quite saw the full connection there, and did a lot of sort of quantum mechanics and statistical mechanics problems. But I've come back to that, and in some sense come full circle. And some of the reason why I actually left doing biochemistry years ago was because of SARS-CoV-1. So years ago, I spent my time as an undergraduate working on the first SARS-CoV, and I'll get to the second SARS-CoV in a moment. But when I was doing that first SARS-CoV, I was actually working on the spike proteins and trying to develop different vaccines against those spike proteins. I realized that just the pace of doing biochemistry wasn't quite for me, wet lab biochemistry, that is. And so then I started to do much more physics and much more computation, and in some sense evolved towards the mathematical dark side. But I've come full circle, and I now realize that the connections among all of these things. And I've started to do much more work in computational biology. And so this is both an old and a new topic in my group at the same time. So that said, today what I'm going to tell you about are really two different stories. The first story is about predicting the viability of beta-lactamase. And beta-lactamase is a protein that many of you may know. It's an enzyme that's responsible for most of the resistance, the microbial resistance that we face in society today. So I'll talk to you about how we can use different measures of biophysics to predict the viability of beta-lactamase. I actually first spoke about this in CCMB about 3 years ago, and now I'm coming back, and hopefully I'll fill in the details and give you the complete story there. The second story that I'll talk about is a more recent story, which has to do with actually creating a machine learning pipeline for predicting pathogenicity. So I sort of view pathogenicity as almost another way of thinking about fitness, but of course the pathogenicity, the harm that different proteins create in one's body as they undergo different mutations. So that said, what I'll do is I'll begin by giving you a little bit about our motivation. Why would we predict fitness, and why would we predict the viability of different proteins and the organisms as a result? I'll then talk about how we actually do those predictions. So the way that we make these predictions of biophysical characteristics is basically through a variety of different types of high throughput modeling techniques. After going through those preliminaries, I'll talk to you about the first story of predicting beta-lactamase fitness, and then I'll go through the second story. We'll see how much time we have for the second story, but the second story really rounds things out. Okay, so why would we be interested in predicting things like fitness and viability in the first place? Well, the primary motivation for this is actually exactly what we're experiencing today. And believe me, I got into this way before we were thinking about COVID-19. The primary motivation for this is trying to defeat a variety of different pathogens. So today, obviously, what's on our mind is COVID-19. And when you think about COVID-19, what we hear on the news basically every day is how many different mutations that are leading to different variants are arising, and of course, stoking fear in all of us that maybe COVID-19 will be able to fend off different vaccines. And so our motivation is trying to think about how can we actually create vaccines, and how can we create drugs that in some sense keep evolution in mind. So if we are able to predict how a particular virus will evolve, then that allows us to figure out well ahead of the virus how we can drug it in a way that's resistant to that evolution. So being able to predict exactly which mutations will lead to profitable pathways, in other words, fit pathways for the virus, is extremely important to being able to drug that virus overall. So I was looking at today, exactly how many mutants have actually been identified in COVID-19. It's well over 7,000 different mutants. Now, obviously, not all of those mutants are fit, but if we were able to say which of those mutants were fit well ahead of time, then we would be able to get ahead of this virus in some sense. The reason why this occurs, if you really think about it, it has nothing to do in particular with COVID-19, but it occurs because of the fact that different types of viruses and smaller organisms can evolve much more quickly than we do. So here's a plot of mutation rate versus genome size for a variety of different organisms. And what you'll quickly see if you're just thinking about mutation rate is compared to us, so I'll group us as higher eukaryotes, things like viruses actually evolve about 10 to the 4 to 10 to the 6 times faster than we do. And so because of the fact that the mutation rate is so high, essentially these viruses are always going to be outpacing us and sort of getting around our different defenses very, very quickly because of how quickly they can evolve. And so what that creates in a more general sense is sort of a whack-a-mole situation. So as soon as you create a drug in the standard pipeline right now for one specific mutant, then all of a sudden another mutant appears, and then another mutant appears, and then you're trying to create drugs and vaccines for every specific type of mutant. And so again, what we try to ask is, in order to combat this situation, can we actually create drugs that are cognizant of all of this kind of evolution, all of these mutants aggregating well ahead of the time that they do, so that we can predict what's fit beforehand. Now, another motivation for this work, of course, doesn't necessarily have to do with just different types of smaller organisms, but also has to do with ourselves. So as many of you, of course, know in CCMB, right, mutations are what distinguish us as different species and even as different individuals. All of us have our own set of single nucleotide polymorphisms that essentially characterize the way that we function. And so another motivation for this is also being able to predict what happens as a result of the mutations within us. And so while some of these mutations we like to think of as being characteristic of us and imbuing us with our positive traits, obviously many of these mutations also imbue us with our negative traits. And if you think about things like cancer and aging, those kinds of conditions tend to occur because of the fact that we're accumulating a variety of deleterious mutations over time. And so another viewpoint on this is that we'd also like to be able to actually predict which of those mutations are going to harm us and which are not, well before we actually see them. And lastly, before I dive into our work, as a chemist, I'm very much interested in this question of pathogenicity and predicting fitness because of experiments like directed evolution. And I find these experiments extremely remarkable and extremely inspiring. What these kinds of experiments do is they kind of put evolution into a laboratory. And what they attempt to do is they attempt to actually evolve different enzymes that will perform specific desired functions for us using an evolutionary pathway. So as this figure on the left demonstrates, what some scientists do, Francis Arnold is one example, such scientists, is they try to take different sequences, they mutate those different sequences, and then they insert them into bacteria, which then produce related proteins, and those proteins perform different functions. And based upon how those proteins function, how well they function, if they produce the desired function or not, those proteins can be selected out. And their related genes can be, the genes that are desirable can be selected in, and then they can be continually evolved in sort of a self-consistent fashion until you produce proteins and enzymes that perform the functions that you want. And so this is incredibly interesting and incredibly exciting for chemists like me, because you can actually create enzymes that do things that regular synthetic chemistry essentially can't in a test tube in a lab right now. So one example I show on the right is just the evolution of an esterase enzyme that has been evolved in the synthetic laboratory. One question about this, about these kinds of evolutions, is just that there's always a trade-off between stability and activity whenever you're trying to evolve these enzymes. And so if you wanted to actually make a better enzyme, you want to be able to predict whether it's going to be more stable, whether it's going to be more active, and whether you can actually create enzymes that are both at the same time. And so given all these motivations, what my group has been very interested in doing is trying to predict fitness based on biophysical properties. So like I said, the biophysical properties that we think about are things related to folding, like the free energy of folding. I mark the free energy of folding as delta delta G, where G is the Gibbs free energy. And delta delta G is basically saying that the folding free energy difference between a wild type protein and a mutant protein. So we're interested in predicting biophysical properties like this free energy of folding, but also things like the free energy of binding. So if a protein binds a given ligand, how well does it bind that ligand? Or if it binds another protein partner, how well does it bind another protein partner? As well as various what I'll call dynamic or kinetic measures. So how fast does an enzyme actually process its substrate? How fast does a protein fold or unfold, et cetera. And so we're trying to use these different measures to predict the fitness of different organisms. But beyond this primary aim, the other thing that we're interested in trying to understand is if you actually know these different biophysical properties based on computation, how well do they match experimental predictions? So we're not quite at the point, I would say, where computation exactly matches experiment. So when we go ahead and we predict these different measures, there is, of course, some error in our predictions. And so we want to understand exactly what are those errors and what are the limits of our computational techniques for predicting these different things. Because as I'll say in a moment, we can spend a lot of time very, very accurately calculating these different properties. But usually that accuracy comes at the cost of a huge computational expense. So typically, we're kind of in a seesaw pattern where we're trying to find the right equilibrium between cost and accuracy. And so what that means is that typically we're settling for predictions of these measures that aren't exactly right. Okay, so the way that we're actually going to get those dots and predict those different biophysical properties is by using atomistic modeling. And so perhaps this is a little bit different than what many of you do in CCNB. But what we're interested in is actually being able to model different proteins and, of course, other materials as well, based upon their atomistic physics. So if you think about atoms in a biomolecule, or in fact in anything, in materials, in regular small molecules, right? All of those atoms have forces, and we can predict those forces using quantum mechanics. If you can sufficiently accurately predict those forces and produce potential energy curves, what you can then do is basically simulate the dynamics and all the interactions among those atoms in order to reproduce the entire function of the system. So on the left, I'm showing you an example of what we think about when we think about atomistic modeling. We're thinking about actually simulating all of the different atoms and their interactions within a protein and with waters around it. For a very, very large system. Now, this is an amazing tool that's really evolved over the past 20 years or so. Of course, people thought about atomistic modeling. Actually, my postdoctoral advisor, in some sense, was one of the first people to do this about 60, 70 years ago. But when they first started thinking about doing this kind of modeling that provides incredibly rich insights into what's actually going on in molecules, of course, they didn't have the computational power to do this. But over the past 20 years, we've really been able to realize the computational power that allows us to really actually model atom by atom interactions for reasonably large proteins and reasonably large systems. So we're no longer in the regime of sort of these very empirical coarse-grained models. We're very much in the regime of being able to go atom by atom throughout these systems. And this has really revolutionized biology, because what we can now do in my group and in others is interrogate exactly what's going on in these different systems. And so our group has been making great use of all these different atomistic modeling techniques. But as I was alluding to before, when we are thinking about how we're going to model different systems, we always have to balance accuracy versus computational cost. And generally, these two things go in the opposite direction. So when you're thinking about modeling things more accurately, often that's accompanied by increasing computational costs. And so when we're modeling these different systems and trying to predict different biophysical properties, we're often choosing between different levels of accuracy and cost. And on one end of the spectrum, what we can choose are what I'll call knowledge-based potentials. These are potentials that allow us to predict properties like the free energy of let's say, folding or binding based upon functions that are parameterized by known free energies for smaller processes. So for example, if I want to predict the free energy of folding of a protein, which basically tells me how likely a protein is to fold in some sense, then the way that I can do this is I can come up with a function that if I tell it, let's say, what's the free energy of a hydrogen bond forming, or what's the free energy of some van der Waals interaction, then based on that function, we can go and we can take all those inputs and we can actually compute the free energy of folding for the entire system. And so there are a lot of methods that do this. These methods are very cheap because they have these different interactions. These free energies are already parameterized. And methods that are like this include things like FoldX and pop music. There are various servers for these things. You know, the saving grace is that we're coming up with increasingly more accurate and increasingly more parameterized functions that allow us to predict these folding free energies better and better. And these things are extremely cheap. So we're able to do things very, very quickly. But as these functions become even more complex and better fit to different data sets, we can do so even more accurately. So on one end of the spectrum, we have these knowledge-based potentials. On the other end of the spectrum, you sort of have what physicists like me love to go to, which are the fully physics-based potentials. So these are potentials that take the XYZ coordinates of all the atoms in your system and most likely also the velocities of the atoms, and then essentially are able to solve Newton's laws, and from Newton's laws be able to obtain a variety of physical properties. These methods are much more expensive, but they're also much more accurate. And so we're always kind of trading off on our seesaw between these two different types of potentials, and these two types of ways of trying to model biophysical systems. But in this work, we'll choose two key techniques. So the first is FoldX. The second is Rosetta, and Rosetta is a technique that sort of sits in the middle here. So I'll get to that in a second. So FoldX, as I was saying, is essentially a parameterized function that allows you to calculate the free energies of folding in it and also binding of different proteins. What it does is it takes in the protein's 3D structure, and then just based upon the coordinates of all the atoms within it, it's able to calculate the free energy interactions, and based upon its parameterized functions, these A, B, C, D's, E's, and F's, it can then get the free energy of the entire protein. So this is a really nice technique. It's very quick and lets us get to the answer with a reasonable amount of computational time. Why do we actually want to do these things in a fast way? Why don't we always stick to the more expensive ways? As I'll show you in a second, if we want to get at the fitness of different organisms, we actually have to scan through all the different mutations of various proteins. And if you think about mutation space is vast. It's a huge space. And so what that means is you have to be able to use techniques that go through that space sufficiently quickly so that you can actually come to an answer about what's going on. The other technique that we'll use throughout this work is a technique that's called PyRosetta. PyRosetta is a Python derivative of Rosetta, which many of you may be familiar with, really came out of David Baker's lab. And what Rosetta allows you to do is it allows you to actually combine a little bit of those functionals of free energy, but also with ways of actually folding a protein. So when I was talking about FoldX, I didn't mention anything about relaxing the structure, or I didn't tell you how we're actually getting the protein structure in the first place. FoldX doesn't actually allow you to figure out the structure. You have to give it a structure. In contrast, PyRosetta allows you to try to figure out what is the best structure within some limits by doing a variety of Monte Carlo moves. So Monte Carlo moves allow you to sample little twists and turns of the different bonds in your protein. And eventually, after sampling all those turns, twists and turns, and determining which are the most favorable, PyRosetta is actually able to come up with a structure which you can then figure out the free energy of. And so in that sense, PyRosetta is sort of a mix of some of those more physicsy techniques that would allow you to figure out things like structures and some of those more empirical techniques. Okay, so given those two methods, I'll jump into my first story, which is actually being able to predict the fitness of beta-lactamase. As I alluded to earlier, beta-lactamase is our lab rat because beta-lactamase is a very influential and also very well-studied protein. And that's because of the fact that the beta-lactamase is an enzyme that's responsible for most of the drug resistance that we see in the world today. What beta-lactamase does is it's an enzyme that very specifically is able to target a variety of what we call beta-lactam drugs. These are really the drugs that started off the pharmaceutical revolution across the world about 100 years ago. So the first beta-lactam drug was penicillin, as discovered by Alexander Fleming essentially 100 years ago. And what these drugs all hold in common is the fact that they have a very interesting beta-lactam ring. So for a chemist, this is kind of a very strange, very peculiar ring, but this is a four-membered ring that you'll see in the middle here. It has a nitrogen and it also has a double bonded oxygen. And this four-membered ring serves as the core of essentially all beta-lactam drugs. But what beta-lactamase is able to do is beta-lactamase is able to actually break this ring. It's able to hydrolyze this ring so that these drugs can no longer function. So what do these drugs do in the first place? Why are they useful? Why are they important? Well, what these drugs were designed to do was to essentially occlude the binding site of what we call penicillin binding protein. What penicillin binding protein does is if there are no beta-lactam drugs around, penicillin binding protein will essentially stitch together the cell wall, so this peptidoglycan glycan wall that's found in many bacteria by basically transpeptidase. It serves as a transpeptidase that connects the different peptides that form that wall. So you see this gray protein typically allows you to actually connect all the subunits that then form a very strong, tough cell wall. But what the beta-lactam drugs do is that they actually occlude the binding site of this penicillin binding protein. Maybe I shouldn't just say occlude. They actually react with the penicillin binding protein so that it can no longer stitch together the cell wall like it normally would. And so when it can't stitch together your bacterial cell wall, obviously the bacteria their walls become extremely weak, and eventually the bacteria will succumb to that weakness overall. Now, beta-lactamase prevents all this from happening. Beta-lactamase does that by hydrolyzing this beta-lactam ring. And what's unfortunate about this situation is that there have been many different beta-lactam drugs developed over the years. So originally, when it was found that beta-lactamase was able to hydrolyze the beta-lactam ring of penicillin, so we see penicillin here at the bottom, of course, scientists tried to develop other types of beta-lactam-based drugs, and of course, other drugs as well. And so one example drug that they came up with was a class of drugs called carbapenems. But soon afterwards, beta-lactamase sort of caught up with those carbapenems and then also cleaved those carbapenem beta-lactam rings as well. And even further on, when we go back maybe 20, 30 years ago, there were also beta-lactam drugs like cefixime, which is a type of cephalosporin, which also were created on the market. And then eventually beta-lactamase was also able to target them and hydrolyze their rings. And so what you see is that beta-lactamase is our lab rat, but also an extremely important enzyme because of the fact that I was able to cleave all these different drugs. It's continually evolving such that there aren't that many drugs that are foolproof against the different types of beta-lactamase mutants that are out there. So given this background of beta-lactamase, what my team decided to do was to actually try to figure out whether we can predict the evolution of beta-lactamase based on different mutations that we can introduce into it. So the key people on my team were Jordan Yang. So many of you may know Jordan. Jordan is currently a fourth-year chemistry and also CS graduate student who's been focusing on different types of folding, binding, and dynamic studies. Jordan did all the computational work that I'm going to present shortly. The other members of my team were experimentalists, also at Brown. So some of you might know Mandar. Mandar is at the EBSCO proteomics facility. And also Dan is a professor in EED who I think is very much involved in CCMB. They were also very interested in trying to create experimental analogs of beta-lactamase with all the different mutations that we were studying. So together, what we tried to do is figure out what was the actual fitness, try to predict the fitness of all the different types of beta-lactamase single mutants that you can make, so that we can eventually predict how they'll evolve and how they'll affect different beta-lactam drugs. So to do so, what we started with was making what we call the Wiley mutant data set. So Scott Wiley was actually a postdoc of Dan's who came onto the scene well before me, made a few mutants, but we actually took off where Scott left off, and we made many more mutants. So we came up with a set of about 15 single mutants and 6 double mutants of beta-lactamase. So single mutants are when you introduce a mutation at a single amino acid. Double mutants are when you introduce 2 different mutations at 2 different amino acids. The reason why we're interested in both single and double mutants is because of the fact that double mutants in particular are interesting because you can imagine that some double mutants would actually be correlated. So if you have a single mutant, right, you only have 1 mutation, you can try to predict what's going to happen as a result of that mutation. But if you have double mutants, you can imagine that sometimes those mutations will counteract each other, or otherwise add together in a way that's greater than the sum of their parts. So for that reason, we were interested in studying both single mutants and double mutants. And the mutants that we focused on were all mutations that were greater than 6 angstroms from the active site. So at first go, we're interested in trying to predict the folding of these beta-lactamase enzymes and mutants. And we didn't want to think much about the binding that's going on there. And so that's why we made all of these mutations reasonably far away from the active site so that we can just see the influence of what's going on with the folding. So we did that, and it was actually heroic. It doesn't sound like a lot of mutations, but that's actually more mutations than anybody has ever made and measured the fitness of directly and the free energies of it in a beta-lactamase protein. I'll get to some other experiments that measured a large number of fitnesses later. But when we came up with this large set of mutants, and then once we have these experimental free energies and fitnesses, we then asked the question, how well can computation actually predict these different free energies and fitnesses? So this was essentially aim 2 if we're looking at the free energy predictions. What you can see on the plot on the left, we're using one type of technique, that's the FoldX plus MD that I introduced before. What you can see is that if we try to predict the free energies that we observe experimentally, that FoldX is okay at doing so. So we get R values of about 0.27 if we use FoldX. But we see that if we actually integrate many more realistic physical interactions into our modeling, that our correlation goes up to something like 0.67. So this isn't perfect, but I also like to look at the high points of this. What you might notice on the plot on the left is that our R value is small, but what we're very, very good at doing is predicting when a particular mutant is stable or not. So the gray regions I show here are when you're sitting in, I'll call it the positive quadrant, when your predicted folding free energies and your experimental folding free energies are both positive. When they're both positive, that usually means that your protein is going to be somewhat unstable. So if your folding free energy, the change in your folding free energy is positive, you're going to be less stable than you were before. On the flip side, if they're both negative, that means that the protein is going to be more stable than it was before because of the mutation. And so what you can see here is that actually FoldX does a reasonably good job of being able to say when is a particular mutant going to be more stable and when is it going to be less stable. In almost all cases, it's able to put these different dots in the correct gray boxes. Pyrozeta can do something similar, but misses a little bit more. But the trade-off is that even though it misses a few more of these, it's actually able to get the overall fit a little bit better because it works on a wider scale of units. So we can get reasonably accurate predictions. That made us a little bit happy. And so then we said, well, what would happen with different types of double mutants? So double mutants were difficult because of what I was suggesting before. If you have double mutants, the mutants can actually interact to be more than the sum of their parts. And this is an interesting story, something that I won't talk about too much today. But what you can see from all this is actually our techniques don't do incredibly well at figuring out these correlations between multiple mutations. So on the left, I show, again, a plot of the free energy of folding of pi rho zeta. So this is the prediction that we would get from pi rho zeta if we ran the calculation on both mutants at the same time. On the x-axis, I'm showing you what we get for the folding free energy if we just added in the free energies from each individual mutation that we introduced into the double mutant. What you can see is that pi rho zeta almost always overestimates the free energy of folding above the combined system. What you can see in the case of FoldX is that even though it looks amazing, it looks great because if you're not thinking about things carefully, everything seems to fall on a line. What you'll notice is that what FoldX is essentially doing is that it's predicting that the combined system is always going to just be the sum of its parts, which we in fact know is not the case, and we can see that in the last plot. The last plot shows our experimental folding free energies if we add them on the x-axis versus if we actually measure the double mutant itself. And we see that if we measure what happens in the double mutant as opposed to adding up the individual contributions, we see that we're definitely not on a straight line. The double mutants have mutations that clearly interact with each other in a strong fashion. And so we see that both techniques, both of our computational techniques, pi rho zeta and FoldX, don't quite get things right for different reasons. So pi rho zeta doesn't get it right because it's just overestimating everything. And FoldX definitely doesn't get it right because it just doesn't even account for correlation among these different mutations. And so this is something that we'll continue to work on, but it's something to be aware of. That said, if we put together our single mutants and our double mutants using pi rho zeta, which will be our go-to method from now on, we can get a correlation of roughly 0.63. So this correlation coefficient is not too bad, but again, it's not perfect. And it's not perfect because we're not using those incredibly demanding computational methods. We're settling for things that are in that computational middle where we're getting a reasonable amount of accuracy also for a reasonable cost. That said, what we wanted to do is then jump to fitness, and it's an interesting story. So when we were thinking about where do we get a large set of fitness data, we thought, we scratched our heads a little bit, and we thought about contacts. And then I remembered that actually one of my high school classmates came up with a way of measuring large amounts of fitness data using relatively few experiments. So here's me back in the day. I'm kind of hiding in the back. And there's Elad Fernberg. Elad Fernberg was one of my classmates. And as part of his PhD in postdoctoral studies, he essentially came up with a way of engineering bacterial bandpass filters that create bacteria that can respond to different concentrations of drugs within very, very specific ranges. And the reason why he wanted to do this is because if you can measure how well, how much they respond to different drugs, then you can calculate their fitness. And you can do it incredibly accurately if you make these bandpass filters, right? So you can basically stitch together different concentrations of drugs that the bacteria can respond to. And if you stitch together all of those little pieces, you can come up with a very, very high resolution measure of what the fitness of all those bacteria are going to be. And so in fact, several years ago, he did that. He came up with a way of figuring out the fitness of large numbers of single mutants. He came up with a data set. And so we said, heck, why don't we use this? Why don't we see how well we can predict this fitness if we can do a reasonable job of predicting free energies? So what we did is we took our free energies and then we calculated all those free energies and compared them to the fitness that he experimentally determined. And then we saw, how predictive are we in determining the fitness based upon our free energies, given the caveat that our free energies are not totally perfect. How well can we actually do? And so what we saw was very interesting, in my opinion. I think is very emblematic of essentially what's going on in the larger field right now. So on the y-axis, I plot Elad's fitness data, which tells us how well a bacteria with different mutants actually responds to different types of drugs. And I plot that against the folding free energies that we were predicting. So here I show FoldX, but we can also do this using pi rho zeta. We can do this using a variety of different means, but the stories essentially remain the same. What you see from this plot that's very interesting is that we're very good at being able to predict when a bacterium is going to have a high folding free energy, and as a result is going to have a low fitness. So what we can easily do is in our techniques, we can figure out when a particular mutation is going to give us a high folding free energy. And when that occurs, we can almost be completely assured that the fitness of that bacterium is now going to be quite low when that folding free energy is high. So if the particular protein that we mutated completely unravels, it doesn't fold anymore because its folding free energy is high, we can rest assured that basically it's not going to have a high fitness. So we do a great job there. Our methods are extremely predictive of that. What our methods are not good at is sort of the opposite situation. So in many cases, what our methods would predict is that our folding free energies are low, around, let's say, 0 or 5 here. So 0 or 5 kcal per mole is not really enough to completely unfold most proteins, certainly not for beta-lactamase. So beta-lactamase sits in a minimum that's lower in free energy than, let's say, 5 kcals per mole. And so when we're looking at folding free energies that are between 0 and 5, what's interesting is that we see a whole range of different fitness values. We see that our different proteins lead to fitnesses that go literally from 0 to 1, and they completely span that range. And so what that indicates to us is that even though we would expect that if the folding free energy were close to 0, that we'd like to see a very high fitness, what it shows us is that actually the folding free energy can't be the whole story. Because if it were the whole story, we would just see at folding free energies of 0, we'd see fitnesses of 1, and at very high folding free energies, we'd see fitnesses of essentially 0. That's not what we see. We actually see this very rich corner around this 0-0 place that we can't quite explain. You know, why would the fitness of our mutants be 0 if the folding free energy changes are very, very small, if the mutation is not actually causing the protein to unfold? And so this got us thinking a lot about other ways of looking at the fitness of these proteins. So if folding free energies don't do it, can something else do it? And so we started thinking about, well, can we actually also look at binding? Can binding tell us whether some of those mutants that fit in that 0-0 corner, can it tell us maybe that there's some other dimension to them? Maybe their binding is very bad, so their folding is good, but their binding is bad. And we can do the same thing with binding. So we can use methods that are very, very similar to Pyrozeta or Foldex to try to predict the binding of ampicillin to these different mutants. And what you see is actually an effect that's very, very similar. So here I plot fitness versus binding free energy. And when I plot fitness versus binding free energy, we again see that there are some mutants that have very, very high, very large binding free energy changes, meaning that they're not going to be good binders upon mutation. But we also see a very large number of mutants that again sit in this 0-0 corner, right? So they have low fitness, but their binding free energies haven't changed that much either. So that means that binding is also not really affecting them. And so putting this all together, we decided to combine folding, combine binding. How well does this do for us? What we see is that if we combine them, we get something that's a little bit better. But again, we're still stuck with this sort of dreaded corner region where we have low fitness, but we also have properties that seem not to be disrupted whatsoever. And so this got us thinking a lot about what's really going on in these different systems. And so towards the end, once I start to summarize, I'll get back to this basic idea. But essentially what we're thinking about is that there are many, many, many different properties, many different features that can affect the folding and binding of these proteins. And it can be hard to hit on exactly which one is causing their fitness to decrease. So when we're showing plots of just folding or showing plots of just binding, in many of those cases, we're actually missing the key biophysical characteristics that would actually predict these proteins' fitnesses. So I'll show you more about this in a moment. Where am I with respect to time? Okay, we've got 10 minutes. So maybe I'll flip through just for the sake of time. Suffice it to say, we spent more time on binding and more time on actually finding mutations that cause interesting changes in binding. But one way we wanted to move ahead with these kinds of ideas, noticing that basically we're not able to make perfect fitness predictions, is that we wanted to try to figure out which features would actually be important for predicting fitness. So if it's not just binding, it's not just folding, what else could actually be predicting fitness? So far we focused on bacteria and thinking about the fitness of bacteria, but we can also think about fitness of human beings as well. And so what we decided to do was actually turn to predicting the pathogenicity of different mutations in real human beings. And to do so, what we did is we took over 20,000 ClinVar entries, and we decided to try to predict whether those entries would be pathogenic or not compared to the data in ClinVar based upon sequence, structural, and dynamical information. So far, when I was talking about binding and folding, that really had to do with structural information. But we said, well, sequence should also predict. That's what people have been doing in computational molecular biology forever. But dynamics should also be predictive. Let's see how predictive they actually are for this relatively large set of different mutations. And so what we did is we gathered up a number of different features. We gathered sequence-based features, we gathered structure-based features, and then we gathered dynamics-based features. Some of our structure-based features were essentially the same, folding free energies. For sequence-based features, we took a variety of different features, but one key feature that we used is something called the variation number, which was originated at Brown. And the idea behind the variation number is it essentially tells you how well a specific sequence is conserved or not. So if the variation number is 0, that means the sequence is very well conserved. If it's not, if the variation number is closer to 1, then it's much less well conserved. So we took a number of sequence-based features like that. We combined them with structure-based features like folding free energies, also solvent accessible surface areas. And we also looked at dynamics-based features, which tell you, how does the protein move? Does it move significantly differently than it did before it was mutated? And then we said, can we predict fitness again? And what's interesting, and just for the sake of time, I'll draw your attention to the plot on the right, is that when we went to make these predictions against the ClinVar pathogenicity data set, what we saw is that actually almost all of these different features contributed to trying to explain the variance in pathogenicity that we observed. So here I'm plotting number of PCs. We took about 15 different features, and almost all of these features contributed in some form to predicting this variance overall. And so that was interesting. But we also asked the question, which of these were actually most important? So as I was saying, we're missing some things when we were just thinking about structural features, just biophysical features. And what we see is that the most important features that I underlined in red were actually sequence-based features. And so maybe to some of you in computational molecular biology might be saying, well, yeah, we've known that forever, but it was kind of interesting to me in the sense that sequence doesn't necessarily tell you exactly what's going on in the protein, but yeah, it was still able to capture whether something's fit or not. So the very fact that something has existed for a long time, has been conserved for quite some time across many organisms, was a better indicator of fitness than essentially anything else. But we did in fact see that a number of other features mattered. It's not like they didn't matter at all. So folding free energies did matter a little bit. Things like mean square displacements, which tell you, how essentially how much the protein shifts based upon the wild type structure. Those things also mattered, but much less than those sequence-based features. And so this was an extremely interesting observation, and it's something that we're still thinking about today. One caveat that I'll put into this is that all these features are calculated in a relatively rapid way, because in order to be able to predict so many different types of mutants, all these features for all these different types of mutants, you need something that's high throughput. So just like in the folding free energy case that I showed you, there are errors in predicting all of these different measures. Nevertheless, you still see these trends. So there's a caveat, but still some of these features are being much more predictive than other things would be. So that all said, I'll just move ahead for the sake of time. And just to summarize, what we've done with our work here today is try to show whether we can use biophysical features to predict fitness or not, and to understand which features are actually most important for predicting fitness. So what I showed you is that we can these days do a relatively good job of predicting folding free energies, binding free energies, and other biophysical characteristics. want to do a better job, we can use more computationally expensive methods, but there are in fact high throughput methods that can do a reasonably good job of predicting these experimental measures. Given those different predictions of biophysical characteristics, we can use them to predict fitness. But the problem is, and the problem that we're stuck with is that many different possible features, many different biophysical characteristics could be responsible for a low fitness. And so we've sort of been stuck, I'll say stuck as a community in trying to think about which biophysical features would be most predictive of fitness overall. And what I've showed you at the end is that after studying a variety of different kinds of fitness, in specific looking at things like pathogenicity, we found that actually sequence-based features are most important for predicting these things, but biophysics matters when biophysics can actually get at a very specific mechanism. So I didn't have time to go into an example that shows you when biophysics is getting things far more right than sequence-based features. But let's just say in specific cases that the biophysics can do better, but in the general case, it's actually sequence-based information that does the best. So just to summarize everything that I've said, I like to think of an analogy. So at the beginning of my presentation, I said that I was just coming back from Cape Cod. You know, here's a van that we went to Cape Cod with, and this van had a check engine light. And I like to think of that check engine light as telling you generally your car is having a problem, your van is having a problem. And so in that sense, you know, sequence-based features, sequence-based information is kind of like a check engine light. It tells you that if there's a mutation in a highly conserved region, that there could be, in fact, a problem. But in contrast, biophysics-based features tend to be much more specific and helpful for you to figure out what are specific things that are going wrong. So they're, in general, they're not going to tell you, if you average over everything, they're not going to tell you that you have a huge problem. But if you know exactly which biophysics-based feature is causing the changes that you see in your protein, or causing, in this case, your car not to work, then that can be incredibly helpful and incredibly powerful. So I like to think about more specific indicators as kind of like our dynamical or structure-based features that can really pinpoint things for you when you need to. But if you average over all of them, they don't necessarily give you a lot of information. So with that, I'll just skip to the end and acknowledge that the key people that contributed to this work. So much of the Beta-Lactamase work was done as part of a larger EPSCoR team. So Dan Weinreich ran our team at Brown, but of course, my group was in charge of all the biophysics-based metrics. And we also significantly collaborated with a team at the University of Idaho, which consisted of a group of modelers who were interested in a wide variety of different proteins. I didn't talk about some of our other protein work. And I'd also like to thank some of our collaborators on the pathogenicity work at Brown. So Neil Starkar was our key collaborator there. Again, Jordan did most of the work for predicting those pathogenicity, predicting pathogenicity using a variety of different features. So with that, I'll thank you all for your attention, and thank you again for coming on such a bright sunny day.
Thanks so much, Brenda. So we have some time for questions, and I actually wanted to start with one of mine, if that's all right.
Go for it.
So this is kind of a more basic question, and I know that you can't necessarily know the exact protein conformation, but when you're looking at these mutations and seeing the possible like delta delta G, the change in folding, does it seem like they're all, you know, like small tweaks, or do you see some that, you know, the only possible outcome could be a very big change to the protein?
Yeah, interesting question. So I didn't hammer this point home for the sake of time, but we do see things across the entire spectrum. So if I look at this plot here, if I see things with very, very large folding free energies, these are often situations where we substituted a very large residue for a small residue. And in those cases, like you're getting at, we do in fact see very, very dramatic changes. So once we start getting to about delta delta G of 10 kcal per mole, that's when you start to see dramatic changes. But the smaller changes are below this 10 kcal per mole. And that's exactly the area where, you know, the fitness is being affected, but it's not necessarily completely deleterious, right? So we're seeing small changes and maybe the protein can still function to some degree according to our fitness metrics.
Cool, thank you. Do we have other questions?
Sure, I can ask a question. So it's very interesting that the sequence features are most predictive, at least for some impressionistically larger trends. Maybe it's very interesting. So either a correlation or some kind of meaningful interpretation of the sequence features that were predictive vis-a-vis the fold, the folded structure. Where are they in the folded structure? Or it's a fascinating question.
Yeah, it's an interesting point. You know, we should go back and we should match exactly when we pick up changes in the sequence, you know, where exactly is that hitting the folded structure? Is it hitting binding? Is it hitting something else? Because I agree with you, there are clear correlations there. In the cases when we were looking at pathogenicity, we just didn't map out all of those connections. But I agree with you. You can use them to go in and then figure out what should it be even more predictive.
Yeah.
Other questions?
Sure, I have a follow-up question to that question, which is very, very open-ended. I just wondered if you could say a little more about the specific sequence-based features that are important to fitness.
Yeah, so the one that worked best for us was really variation number. And so I'm not saying that we use an encyclopedic number of sequence-based features, but what we really just needed to see is essentially how frequently did we see changes in a highly conserved area as a result of our mutations. So if it was just one mutation, right, you didn't necessarily count that. But if there are multiple mutations and they're all in highly conserved areas, that was the most important metric for us. And so different features can get at that in different ways. You know, that's also information that we want to disentangle in the future. But it's really for us, when we did our studies, this variation number was quite powerful.
Thank you.
I think we have time for one more question if there is one. All right, well, thank you so much, Professor Rubenstein, for joining us and sharing your research. It was cool to hear about protein folding, and for everyone else, have a good evening.
Thank you, everyone.
Thank you.